JPT is pleased to introduce PepSup, a highly standardized assay format for immune response monitoring. Monitoring T-cell responsiveness to infection, vaccine antigens or tumor antigens, for example, in one or a few study patients can be done by short-term activation assays such as intracellular cytokine staining or ELISpot, to mention the two most common ones. However, for monitoring large numbers of individuals, the traditional assays are too complex. While typically antigen-specific T-cells are monitored clinically by ELISpot or intracellular cytokine staining, the routine application of these methods for hundreds, thousands or even tens of thousands of patients will be too labor-intensive and costly. Also, as shown by a range of studies, by using ELISpot or intracellular cytokine staining by flow cytometry, it is very difficult to achieve the level of standardization and reproducibility that would be required for making results from different centers comparable. This is down to the fact that there are too many steps in these assays, too many variables, as well as instrument variation at the point of sample acquisition. Monitoring needs to be simplified without losing sensitivity or specificity. The use of clinical immunotherapies targeting T cells in an ever-increasing number of patients, in particular cancer patients, raises the question how we will be able in future to monitor the success of such therapies at a reasonable cost. Clinical monitoring of immunotherapy in large numbers of individuals is a significant challenge. It needs to be sufficiently sensitive, specific, and effective, and it must be affordable. One way of doing this is to monitor the cytokines or other activation markers produced and secreted by activated T cells in culture supernatants from whole blood. Avoiding PBMC preparation saves time and eliminates sources of experimental variation. For this approach, the same peptides can be used that would normally be used in ELISpot or intracellular cytokine assays. It is an advantage for any study that ELISA and similar formats are easier to standardize than ELISpot or flow cytometry. Thanks to new, highly sensitive commercial assay formats, secreted cytokines have become a more sensitive readout than in the past. These assays can be standardized to a very high level. Even small numbers of activated T cells can be detected. Multiplex readouts can be used. Where a single cell resolution is lost, the composition of cytokines may provide additional clues as to the T cell subtypes and other immune cells involved in the response. Typically, in order to stimulate whole blood or PBMC with peptides, these peptides are first dissolved in a solvent to produce a peptide stock solution and are then further diluted to provide a working solution, usually at the time of the experiment. They may also be stored as aliquots of the working solution. For activation assays, aliquots are added to whole blood or PBMC prior to incubation. Dissolving and aliquoting peptides are major sources of error and experimental variation in clinical studies. PepSup can make your life a lot easier. JPT can provide ready-to-use tubes containing enough peptide for one test in a special peptide incubation solution. You simply have to add whole blood to the tube, move the tube gently to allow mixing, and incubate. The incubation solution does not require CO2 for attaining the correct pH value. Following incubation, the tubes are spun and the supernatant is collected and stored for analysis. Along with the tube containing the peptide antigen of interest, JPT provides a negative control tube containing all the same reagents except the peptide or peptides of interest. We also provide a positive control tube containing a positive control peptide pool. For performing the assay, the same amount of whole blood is added to these tubes and they are incubated in parallel. One of the big advantages of not requiring CO2 is that incubation can take place with tightly closed lids in a heat block. Ideally, a heat block is used that can also cool down samples to 4 degrees Celsius after the desired incubation time, so that further sample processing can be delayed by a couple of hours with no disadvantage. This is particularly useful if incubation ends in the middle of the night, since the supernatants can be collected in the morning. Supernatants can be analyzed immediately or frozen for later analysis. They may be analyzed with respect to cytokines or other mediators of interest using ELISA or similar technologies that also lend themselves to high levels of standardization. PepSup is therefore the ideal basis for monitoring antigen-specific immune responsiveness in a range of situations, for example infection, transplantation, or tumor-specific immunity. Because of its high degree of standardization and hence reproducibility, it is particularly well suited for longitudinal participant monitoring. While PepSup was designed for use with whole blood, it may also be used with other cell suspensions. We would like to mention that the PepSup assay format can also be used for intracellular cytokine staining if desired. However, this will be less easy to standardize as a study readout for the reasons stated before. It may, however, come in extremely handy if you wish to run an ICS sample in parallel to your PepSup assay. All you need to do is add a vesicle transport inhibitor at the beginning and then incubate. After incubation, you may proceed as you normally do with ICS samples. JPT can provide single tests worth of peptides in alternative formats upon request. Please contact us for further information. If you have any questions relating to this presentation or any JPT product, please contact us at peptide@jpt.com. Thank you very much for your interest.